I want to start by giving my perspective of vaccine development in the 21st century. Specifically, when I go to meetings, I hear that all the easy vaccines have been done. And I actually think this is the wrong way to think about vaccine development. And I like to quote Einstein's approach to this, and he says, we cannot solve our problems with the same thinking we used when we created them. What I mean by that is first, that our immune systems crave whole bugs. They grew up on those, and our first vaccines were whole viruses, inactivated or attenuated, whole bacteria. And we did a good job. I don't mean to discount what was done in terms of creating subunit vaccines and proteins and polysaccharide conjugates. But if we're going to solve these other problems that we have in emerging and persisting diseases, we're going to have to move to another generation. And I believe that next generation has to do with synthetic particle vaccines. And we heard a great talk yesterday by Jeff Ulmer about his vision of how synthetic particle vaccines are part of this. And so what I'm going to talk to you a little bit about is how we can create particle vaccines where we incorporate proteins plus or minus adjuvants, where we can actually create what we call virtual conjugates, so avoid the chemistry that's involved in polysaccharide conjugates and mix proteins, polysaccharides with particles. And indeed, as Jeff discussed yesterday, the ability to put nucleic acid into synthetic delivery systems. I won't touch on this today because he did a fantastic job on that. So my objectives are fairly straightforward. To talk to you a little bit about the particle features that influence immune responses that we've learned over time, and there's a great literature about this, that relate to size, shape, composition, surface features, surface exposure, and even charge of particles, although I won't have time to talk about charge. I'm going to talk about what we do at Liquidia in terms of manufacturing. It's called PRINT. It's just an acronym for how we do it. And I'll review some immunologic data we have from laboratory animal studies, and then I'll summarize what we've done, and I'll get that done in our 20 minutes. So if you don't believe the data I show you, there's a lot in the literature. This slide is adapted from Gregory and colleagues, and you're welcome to look this reference up. But basically, the hypothesis in this paper is that, and the data that they review, looks at the fact that particles at least 500 nanometers and larger, maybe 200 nanometers and larger, tend to be processed through dendritic cells to a CD4 pathway to the generation of antibodies, whereas particles that are much smaller, in the less than 200 nanometer range, tend to be processed in a way that brings them to MHC1 presentation and CD8 responses. Now, so that's a little bit about size. What about surface features? Again, in the literature, this publication by Vane et al., they took particles, in this case 5-micron particles, that were either spherical and relatively smooth on the surface, or they created buds on the surface, and I hope you can see these little blebs. These are polystyrene particles. And then what you see in these photographs is the association of these in vitro with a macrophage population. And what they did was analyze the association. This is an example of a particle that was internalized. So the red is the particle, the green is the cytoplasm of the macrophage, and the blue is the nucleus. This is an example of one of the spherical particles, which was generally associated but actually not internalized. And then this section, or photograph C, is actually how these blebbed particles actually attracted multiple macrophages. And they did a formal analysis looking at the percent bound particles, and indeed showed that particles that have very rough surfaces, if you will, were bound more highly, about 80%, about fourfold as particles particles were bound to these macrophages as were these spherical smooth particles. And indeed, if you looked at the number of cells that tended to aggregate around a particle, it was a much higher and statistically higher percentage of particles that had more than one macrophage associated with it in vitro. In addition, while the binding and the association is reasonable, they also went on and looked at the ability or the production of IL-1 beta by these cells, and found that indeed, as you got to ratios close to 1-to-1, that you were getting higher levels of IL-1 beta associated with macrophages that were seeing the budding particles. So not only were they associating, but they were actually stimulating the cells in terms of responses. Whereas the spherical particles, while they had some impact, clearly a less significant impact. So this brings us to what I'm going to talk to you about, how we look at particles, and basically our solution for particle design is that this manufacturing process actually allows us to independently control 5 different parameters. These parameters are size, so we can go from small nanometer, 30 to 40 nanometers, up to micron size. We can create various shapes and importantly, a uniformity of these particles through the process. And you see examples here, including actually particles that look very much like pollen, which is actually something that's very important when you're talking about respiratory delivery. We can also surface functionalize particles, so if you need to target, and if we think about cancer, that's an opportunity. If you want to target a surface, you can actually add something to the surface of these particles during manufacturing. And what we found over the course of studying a number of drugs, a number of proteins, including enzymes, is that the process is gentle enough that it doesn't adversely affect the chemical properties of what we're putting in. And then lastly, we can also affect the surface of these particles. We can create pores within the particles and actually make particles that are flexible. So if you're thinking of things in the circulation that may have to get through small capillaries, we can adjust the chemistry to do this. Now, the place we do this and where we do this is one of our manufacturing streams here. To give you a sense of the size, we could fit at least 4 and maybe 6 of these in this room, and I'll come back to that a bit later. And rather than walk you through the individual steps, what I have next is a video narrated by our founder, Dr. Joe DeSimone from the University of North Carolina.
Print is a templated nanofabrication process that builds on advanced technologies from the microelectronics industry and uses it uniquely to make particles of controlled size, shape, and chemical composition. So it's a roll-to-roll manufacturing method. And we start with master templates, where we make the patterns using the advanced lithographic techniques from the semiconductor industry. From that, we developed unique materials we refer to as FluoroCure that we're able to spread on these surfaces. It wets every nook and cranny. We photochemically cure it, and we make molds from these master templates. And then what we're able to do is to fill the cavities with any material that's going to become the precursor to the vaccine or medicine. And we basically marry the filled mold with a harvesting film, pull all the particles out of the mold, and gives us a 2-dimensional array of particles on a harvested film. And then what we're able to do is dissolve away the adhesives used to pull the particles out of these molds, and that gives us a dispersion of our final product. So we're now able to give calibration quality particles and apply them for the first time to develop safer and more effective vaccines and medicines.
And what I'll add to that is what you saw is kind of what we do at the benchtop. What we do in that machine in this roll-to-roll technology is essentially take what can be kilometer-long lengths of those molds and run those through a process where we can fill in batches the material into the molds and then harvest and move forward. So what do we do in vaccines related to this? There are a number of particle design strategies that we use. I'll talk a little bit about each of these. We can, and this is sort of your key over here to the various colored elements here. But basically, we can associate an antigen with a particle without anything additional. We have developed single adjuvant delivery systems, so if we've got a TLR agonist where that TLR receptor is on the surface, then we tend to put our adjuvant on the surface of the particle, add our antigen to that, and then use that. If we're more interested in intracellular stimulation, we can actually incorporate adjuvants within the matrix of the particle and then add our antigen to the surface. And parenthetically, the addition of these can be chemical, can be trapping, can be electrostatic. So there are a number of ways to do this and still preserve the integrity of each of the components. And then we've moved on to more combined adjuvant delivery, where we take this particle with adjuvant internally, add some adjuvant on the outside, add our protein and test that. And we've even tested, in part for control, but we've seen some interesting data where you just add two different particles but simultaneously inject. So it's one thing to create all these formulations, but then we have to go back to that literature that says, you know, shape matters, size matters, conformation matters. So we had a little bit to do in terms of deciding size and shape when we looked at influenza hemagglutinin. And so we tested a variety of sizes and shapes here. And basically what you'll see, and this is in animal studies looking at HA responses in ELISA, is that our cylinders and cubes, which look essentially like this, and they basically have equivalent dimensions, gave reasonable but not the best responses. When we increased the aspect ratio, in other words, made the particles longer than they were wide, we got much better immune responses in these animal studies. But these large particles, if you're talking about eventual manufacturing of a product, are difficult to sterilize. So we actually need to be looking at things we can sterilize. And these particles, small particles, are an advantage because of the ability to filter sterilize. So we went on with the F protein of RSV to look at the size and shape at the small scale matter rather than at the large scale. We saw that it mattered at the large scale, and you'll see here, again, with an ELISA readout, looking at various sizes and shapes. These are spheres of different sizes, and these are rectangular or rod-shaped elements. That in terms of the antibody response, it actually didn't much matter when we were below 200 or 300 nanometers in the size of these particles. But having said that, that's not the whole story. Whoops. Okay, we're sorry for the inconvenience. Let me try this again. Oh, okay, all right, let's see. This worked perfectly in practice rounds. Okay, here we go. PowerPoint and Mac, they just keep not getting along, if you know what I mean. It's Bill Gates fighting forever. Okay. So this wasn't the whole story, and I'm not going to show you CD8 data, but I will show you the CD4 data. And that, as I said in the previous slide, when we looked at various sizes, now in this case we simplified this and only used spherical particles, and we didn't see much of a difference in the ELISA response. So it's various sizes, and for IP reasons, I can't tell you exactly what sizes. But we saw a difference, didn't see a difference in the ELISA titers yet. In CD4, TNF, and IL-2 responses with this antigen, we saw that actually there was a peak size that was important in terms of maximizing the CD4 response as measured in these assays. And indeed, we also saw this, although don't have time to show the data, with HIV. I mentioned another level that we could look at. Those studies, by the way, were all done just with F protein with particles, no adjuvants involved, no aluminum involved in these studies. But we are interested in whether we need adjuvants, and if we need adjuvants, what's the best way to do this? So this is a relatively busy slide, but we're looking at the malaria circumsporozoite protein in these animal immunogenicity trials. And you heard earlier that CSP doesn't do well in a vector. So what are you going to do about that? What's been done to this point has been to use adjuvants. What we found in these studies is while we get a response to CSP in animals that was reasonable, just by having it on a particle, we could improve that about 11-fold. And these sets of data just show that adding either MPL or proprietary TLR7/8 to the particles, to CSP, brings you back, but not quite to what you could do with just particles. And adding a single particle or having a single adjuvant with the particle and the protein didn't really get as much more than the particle itself. But interestingly, when we moved to using both the TLR7/8 and the MPL, and there's an error here, this should be CSP plus TLR7/8 plus MPL, we actually found that we could get somewhere between 40 and 50-fold better antibody responses with that mixture than we could with CSP alone. And interestingly, if we had one particle that just had the adjuvant combined and co-administered with the particle carrying the protein, we still got an enhanced response of about 40-fold, not statistically different than when we put everything in one particle. So there's flexibility here. And it's not just a response in antibody titers, but also in CD4. And here we're just showing you data on the TLR7/8, but we saw improvements in interferon gamma, IL-2, and TNF responses in CD4 cells when we had the CSP and TLR7 in the particle relative to either the soluble with the TLR7/8 or the protein by itself. And this graph on the bottom right just combines all of those data to show you the summary of how much better the CD4 response is when we use a particle in this. Another area that I mentioned is the construct of, I guess you can call this combination vaccines, but more importantly, what we call virtual conjugate for polysaccharide responses. As you may know, in Prevnar and Synflorix, the basis of that is to chemically link a polysaccharide, illustrated here in these different squiggly lines, to a protein. That process can be very inefficient and labor intensive. So what we have done rather is to take a core particle and then again absorb to the surface with a little bit of chemistry a carrier protein. In this case, we're using a common protein in pneumococcus called pneumolysoid, combined with the polysaccharide. And we make each of these, the one for polysaccharide 1, 5, and 14 independently, and then combine them into a trivalent formulation. And when we do that, these are the kinds of data we get in mice. So our gold standard here is Prevnar. And these are ELISA antibody responses, but I will say we've measured functional antibody in terms of opsonophagocytic responses, and they parallel very well, just as you would expect. The ELISA predicts a functional response. And what you see is the time course here of a 3-dose schedule with Prevnar and the response to polysaccharide 1 compared to what we're seeing with the print particles. And these are with equivalent levels of polysaccharide. So there's a 2 microgram dose of polysaccharide of each of these in the final formulation. And we see across all 3 serotypes either equivalent or better, perhaps a little faster response with our particles. Interestingly, when we look at protein responses, so we look at the carrier protein, and one of the reasons to use a carrier protein here is it may give you broad protection against the serotypes not included in your eventual formulation. What we saw is that we had a certain response if we just injected the pneumolysoid, and that when we looked at the pneumolysoid response in the combined particles, it was actually slightly lower over time. And this has been consistent, and it's actually consistent with what's been found in conjugate vaccines in the sense that the carrier proteins in most conjugates aren't as immunogenic as a carrier protein. The antibody response seems to focus more on the polysaccharides. So we believe that actually adds to the sense that we've created essentially a conjugate vaccine. Now, the proof will be when we get into the clinic and we get this into infants, but we're very encouraged by these data. So this has been a little bit of a whirlwind, and I hope I've shown you that size, shape, surface features, surface exposure, composition in terms of density, and I didn't talk about density, but this is an important issue. How dense should each particle's antigen content be? And charge, again, I didn't talk about this, but bottom line there is you need a positively charged particle for your immune system to want to care about it, which doesn't surprise you when you think of how bugs avoid our immune system. But in any event, these are considerations in any particle design, and anyone that talks about particles that they're using to drive an immune response. And I think I've shown you that our technology offers an ability to precisely and independently control a number of these characteristics through the unique manufacturing process. And lastly, I'll just mention that this particle fabrication is relatively efficient and actually has the potential to be to replace or eventually some much more complex manufacturing processes and even facilities, improving scalability and importantly affordability, which is, I think, one of the reasons why the Gates Foundation actually was an investor in our company, while improving the effectiveness or at least having equivalent effectiveness and perhaps improving safety because you're not having adjuvants just spreading out through the circulation of next generation vaccines. So with that, I just want to acknowledge this is our team back in North Carolina, and in particular, I want to thank Drs. Galloway, Relay, and Stone for their assistance in generating and putting these data together. I should also mention the Gates Foundation and PATH for funding some of these research projects, and with that, I'll stop, and thank you for your time. And I'll take some questions while they change over. You can come and change over for the slides. Yeah, sorry.
Yes. On the second slide, you showed the different shapes of HA. Have you compared that directly to flu? So if you have
Sure.
The standard vaccine versus
right.
Yeah, the standard vaccine versus that
Particleized
particle
vaccine.
Size. Where does that fall?
Right. So we've done this one with purified protein and also with Fluzone off the shelf. And indeed, in mice, you get much actually better responses. Now, we actually took that into the clinic, and everyone knows mice lie. Indeed, mice did lie in terms of the ability of the particles to enhance in a previously immune population that was between 20 and 70 years of age. So this doesn't work in all cases. Flu is a little different bug. One thing I will say is that when we did that clinical trial, we just took Fluzone off the shelf and absorbed it onto the particles. And one of the things we think retrospectively is why the mice fooled us is that we may have effectively diluted individual antigens by having all three antigens associated with one particle, because a cell that looks at one particle is probably looking at one of those antigens, and other antigen is being lost and not processed properly. So if I were to go back and get into the commodity market of flu, I might absorb each antigen individually, and I would predict that we would do better than Fluzone there. Thank you for the question.
Did your size and shape dependency hold for larger species? Maybe this is related to the first question. If you didn't optimize the size and shape beyond mouse, maybe that's why you didn't see the effect with flu.
So we did some optimization in using an aspect ratio of about 2.5 to 1 in those particles. We wanted to have a filterable particle, so we used an 80 by 320 particle. And again, the data in mice were showing us that that was going to give us a reasonable response. But I will say a lot of these data relate to studies in animals, and we're going to need to understand when we get into people how much those size and shape issues matter and whether we have animal models that predict that. But it's a good question. Thank you. Yep, sorry.
Just to follow up on the Prevnar study, did you include aluminum with your particles when you compared it to Prevnar?
I didn't show that, but we have additional data where we're actually looking at Th17 responses, and we actually found that we can get Th17 responses. And while aluminum doesn't impact our ability to get an antibody response, it actually enhanced our ability to get a Th17 response. Okay, so do we need it or not? We didn't need it in these studies, whereas, as you know, Prevnar is aluminum-based, but there seemed to be an impact, and we don't understand why yet, of aluminum on the Th17 response, which typically you don't see with these conjugates. So, but I just didn't have time for that. Sorry. Yeah, no. In the RSV data, we did look and saw IgG1. I'm sorry, I don't have the data for that with me to show you, but we haven't routinely done that. We certainly didn't do that with flu, and I don't think we did it in the malaria program.
Basically, do you think that you can manipulate the T cell polarization by using the nanotechnology without adjuvant? Or,
without
adjuvant, just nanotechnology is enough to manipulate the T cell response, such as Th1, Th19, 19, 17 cells.
Okay.
Is it possible?
I'm sorry.
Is it possible to manipulate T cell responses, such as Th1, Th2?
So there are some data in the literature to suggest that, and we've done some preliminary Th1, Th2 type analysis with RSV. It was relatively balanced with just particles, but if you wanted to drive it in a certain direction, then it was important to add an adjuvant that would help drive in that direction to have an overabundance of Th1 versus aluminum, which was giving you Th2. Now, I'm sorry, I don't have time to go through those data. Yeah, I'm going to keep asking questions because our moderator has disappeared, but somebody can stop me with the next presentation. I don't want to slow things down, but go ahead.
Thank you very much.
Oh, okay. So
I would like to ask you to absorb the antigen or adjuvant to the nanoparticle you presented here. Linkers are needed or absorbed?
So yeah, the great question. So the question is, do we need a linker to absorb? Yes. And the answer is not necessarily. We use certain compositions that will associate. So you can imagine lipids on the surface that a lipophilic molecule will stick to. But you can use also charge. So these particles, you'll hear if you're at many of these nanotech meetings about zeta potential, right? This is just whether it's positively charged or negatively charged. And you can, depending on that matrix material, that red box of material, you can adjust what you put into that. You can adjust the positive charge, and then with that positive charge, you can absorb and very tightly negatively charged molecules. So it depends. In a lot of what we do with the polysaccharide conjugate, we actually do gently treat that final particle with a bit of formalin-like material that cross-links, but not in a defined linkage. The protein, and just gives it a little, what we call stitching. It just makes it a little sticky, but not sticky enough to change, or not rough enough to change any of the antigenic properties of either the protein or the polysaccharide.
Thank you very much.
Okay, sure. It's time. Okay, so I don't know who the next speaker is, and I don't see Malcolm. Oh, here we go.